anyway, I'd like to start with the real topic of today, and that is a reappraisal celebration of Schrödinger's book. I did read it when I was an undergraduate. I remember being, I enjoyed it. As Peter says, you read it very quickly, you enjoy it, you understand it. I don't remember it having enormous consequences about what I did, but I do remember having read it and found it very interesting. But I can't say it was hugely influential. Now, going back and rereading it a few weeks ago, it seemed to me that there were three key points that Schrödinger was really trying to make. Well, these are the 3 points that I thought were more important. First was that by maintaining their identity through long periods of time, living organisms defy the second law of thermodynamics, and we've heard a bit about the discussion about that. Secondly, their ability to do so depends on just one copy of a hereditary material that must be an aperiodic solid or molecule. He did use the word crystal, but he also used the word solid. He didn't talk much about polymers, which is strange, I think. But I think one gets sort of hung up on the crystal thing because it kind of sounds nice, but he didn't just talk about crystals, and he didn't dwell on the crystalline nature of it. As far as a physicist was concerned, a large molecule was a solid. And lastly, the ability of an aperiodic molecule to create so much order may or may not, and he was very ambivalent about this in the book, require a revision of the law of physics. I think he very much hoped it would, and we've heard very nicely from both the talks this evening that I think he was probably very disappointed that the truth was much more prosaic. And I'll say a little bit more about that later. I just want to consider these 3 points before I start talking about chromosomes. The first was that he really dwelt on this whole business of homeostasis, this ability of living organisms to defy the second law of thermodynamics. I won't go into how they do so. That's not my area of expertise. But it did seem to me from rereading the book that the one thing that Schrödinger had kind of sort of missed the point, which Dieter made in his little introduction, was the fundamental thing about life is not homeostasis. We've got machines that can do that. That's not really something that's really unique to living systems. What is absolutely unique is self-reproduction. And rereading this book and thinking this through a little bit and sort of seeing the big picture, and this is an audience that maybe would appreciate this, I think that once machines are developed that can reproduce, we as human beings are in trouble, and it will happen, and we will be in trouble. Because evolution is a consequence of reproduction, not the other way around. Dawkins did not understand that. Once you reproduce, evolution is, as Karl Popper pointed out, a mathematical deduction from reproduction. Popper got highly criticized for saying the theory of evolution is largely tautological. What he really meant is once you accept the premises, then evolution must happen. There is no argument about it. It's 2 + 2 = 4. So evolution is a consequence of that reproduction. Once machines start reproducing, they will start evolving. And it probably won't end well, is my prediction. So so now let's go to how did Schrödinger beam in on the aperiodic solid? Well, he beamed in because of this beautiful work that Dieter has touched on, the wonderful work of cytologists in the late 19th century that essentially had worked out that chromosomes were carrying the hereditary material. And really one of the key findings was obviously the description of what happened to the contents of the nucleus. Essentially, cytologists had shown that upon fertilization, it was the nucleus that contained the information, that upon fertilization, it was the nucleus being contributed that determined the character of the development of the embryo. So then people were interested in of the nature of nuclei and how were they reproduced during cell division. And then there was an enormous argument between those people who said, well, nuclear reproduction, which goes hand in hand with cell reproduction, was similar to cell reproduction, namely the cell got bigger and bigger, and at a certain point it divided in two, and half went this side and half went that side. And people say, well, nuclei do the same thing. They get bigger and bigger and bigger and then just split into two. And that was called the direct school. And there are other people, the indirect school, said, well, actually, no, nuclear division is quite different. Basically, the so-called nucleus in interphase, when the cell is growing, the nucleus is growing, you can't really see very much. And then suddenly, before the cell divides, the contents of the nucleus, as it were, crystallize out into what we now call chromosomes. And then they undergo this wonderful dance and this extraordinary segregation process that was described in great beauty in 1879 by Walter Flemming, which we now call mitosis. And this was called, so they said this was called the indirect nuclear division. So there's a big argument. The big difference between those who are for direct division versus indirect is that these guys had better microscopes than the other guys. It was quite as simple as that. And this was largely from the German school. And there were people like Huxley who had completely useless microscopes and couldn't see chromosomes. Anyway, so now this paper is an absolute classic, this description of this process. But the remarkable thing about this paper is there's no mention of why. You know, this happens, but Flemming, there's no mention in his paper of why did the nucleus do this. And I think and just here, okay, I just want to, again, this is a little movie of this mitosis. You can see the chromosomes in dark and the spindles pulling the chromosomes. This is this process of mitosis. It's just an extraordinary, it's the most dramatic thing a cell does. And we're going to go into the mechanics of this later on. Okay, now, so there was only a few years later, and this is one of the very rare papers on theoretical biology. There are incredibly few examples where people have written a paper that just has ideas that has enormous influence in biology. And this was one of them, written by Wilhelm Roux in just 4 years after Flemming's description of mitosis. And he started to ask the question why this very specific appearance of chromosomes, each of which had sister threads. They were Phaden, they weren't called chromosomes, and they had sister Phaden, and they were lined up, and then they moved to the center of the cell, and then they went to opposite sides of the cell. Why this extraordinary choreography, as he drew here? And in this paper, he posed this question, why have a complex process with a simple division would just do the job? And he was sort of clearly groping his way in the dark to getting the idea that what was involved here was the propagation of information. So he talks about the mitotic figures are the mechanisms which allow the nucleus to be divided not only quantitatively, but also according to the mass and nature of its individual qualities. This is the first notion of information being propagated here, and that chromosomes, this process of mitosis, is about the propagation of propagating individual qualities, not just stuff. So that was a very important step in the whole evolution of chromosome theory. Now, it was really Theodore Boveri who did the most beautiful experiments, who really proved that we've all got lots of chromosomes, and in fact, we've got 46. We only need 23. You only need one set of chromosomes, essentially, for life. But at the time, people thought, well, why have 23? Why couldn't you have just one? And there were a lot of people who just felt, well, that one may contain all the information, the other is just sort of backups. And there were very good reasons for this. So, for example, in Britain now we have a terrible problem with Indian mugjack. It's a deer that was imported from India by some idiotic aristocrat in the last century, and it's now a complete pest. Well, the Indian mudjack has only got, I think, 7 chromosomes, but it's got a Chinese look-alike. They look absolutely identical, but it's got 22 chromosomes, okay? And people could see that. They could see that organisms had very similar numbers of, they were very similar biologically, but had totally different numbers of chromosomes. So people said, how could the number of chromosomes be important? It's a reasonable conclusion from that observation. Well, it was Boveri who showed in sea urchins, through experiments I can't go into because they were really quite sophisticated, they were extraordinarily sophisticated, but essentially by programming systematic chromosome missegregation. He showed that you could only produce a living embryo of a sea urchin if it inherited a complete haploid set of chromosomes. This was 1902, just the same time as Mendel's laws were being rediscovered. And Boveri and Edmund Wilson's student Sutton put 2 and 2 together and said, wow, the chromosomes, which we know you need a whole set for development, and Mendel's determinants, maybe these are carrying Mendel's determinants. And that was the birth of the chromosome theory of heredity. It's interesting, it took quite a while for it to be proven by Morgan and his school. The reasons for that are very interesting, but I don't have time to go into that. So that's where Schrödinger is starting from, that he knew, and we knew very well at that time, that chromosomes carried the hereditary material, and that there were essentially one copy was sufficient for life as such. And so therefore the argument that he really made that got him to this aperiodic solid or crystal or whatever was accuracy is in determining the behavior of gas is determined by the square root of the number of molecules, which means that if you've only got small numbers of molecules, the accuracy is going to be very poor. This has been mentioned by Robert already this afternoon. Irradiation experiments by Bellbrook and colleagues had suggested the mutational target within genes was no more than about 1,000 atoms. Now, I suspect you go back and look at those calculations, they were very back of the envelope. There could have been enormous, these numbers with a real pinch of salt. But the key point was and it wasn't the gene. They didn't conclude the gene was 1,000 atoms. It was the unit of mutation, not the gene. And of course, we now know genes can have tens of thousands of base pairs, and the unit of mutation is a single base pair. But the key point was the idea that here was something very small from the point of view of a Boltzmann physicist, and yet once it happened, it could be maintained in a very stable fashion, virtually forever and ever. And so that was really the conundrum, and I think this was the real point of Schrödinger's book, was this sort of how on earth can something so small be propagated so stably for so long? And that was really the thing that got people interested and excited. All the rest of it, a lot of it was wrong, because most of the stuff we know is wrong at any one point in time. But the problem that he pointed to was absolutely right, and I think that's why it was so influential. His solution, this was just a sort of shot in the dark. The unit of mutation must be part of a much larger molecule or solid or crystal, and these words come and go in the book. And because this solid must encode such a huge amount of information, it must be aperiodic. I think that was the basic argument. The other conundrum he had was, how can just one copy of this substance that does not contain very many molecules create so much order? That was his other conundrum, and that's what led him to ask, is this consistent with the laws of physics, and if not, might fundamentally new principles emerge? And I think we've heard already this evening that he secretly, I'm almost certain, did hope that would be the case, and probably was deeply disappointed that it wasn't. So I just now want to mention a little bit of how, from the chromosome, we actually got to DNA. Because we hear a lot about Watson and Crick, but actually what Watson and Crick did was taking the low-hanging fruit. The key point was to get from the chromosome to knowing that DNA was the hereditary material, because chromosomes would contain DNA, which at the time was thought to be a rather simple molecule, and lots of proteins, which was known to be a very complicated set of molecules. And most people thought it was the proteins that must be containing the information. And this is I think, frequently a forgotten paper. It was performed with the lowly liverwort, which is a sort of a primitive plant. It produces sperm and it eggs. And a group in Berlin, I think, back in 1939, so this is the 90th anniversary of this very crucial paper. They then said, already Muller and colleagues had shown that irradiation can induce mutation, and they asked, and it was known also that the spectrum of, the absorbance spectrum of nucleic acids the maximum was around 260, and that of proteins around 280. So they did this incredibly simple experiment, is to irradiate the sperm, and they had to use sperm because you had to have a cell that wasn't going to absorb a lot of the irradiation by other pigments were there, and the sperm were very small, and there were no other pigments, so you could really know your radiation wasn't being sort of absorbed by other things. And they did it irradiated with different wavelengths, and then looked at, first of all, how effective was the irradiation at killing the sperm, and what they found was, in fact, the 260 was much more effective than 280. So this was the viability with 260, and this was the viability with 280. But more important, that they looked at the number of mutations that were produced, and again, this peaked coincide with 260 and not with 280. And so this was really the first paper, and they clearly draw the conclusion that DNA might be responsible for the hereditary material. I think this was an extraordinarily simple experiment, and it's not mentioned very often. Well, from there, it was only another 5 years before this next absolutely crucial paper, and it's very interesting. This is the 75th anniversary of Avery's demonstration that the transforming substance that was capable of transforming the pneumococcus from this non-virulent to a virulent form, which could be seen by the morphology of the colonies. It would be, well, it's been described that you could take an extract from these ones and mix it with these ones and transform them. And once they had been transformed, you could then take extracts from this and do the whole thing again, so it was transferable. And what Amory did was just to take this extract and fractionate it and ask what was in it that was doing it. It was incredibly simple, but very, very technically difficult. And he purified and purified and purified, and at the end of the day, these were the results, and as far as he could tell, the substance that was responsible was the DNA and not the proteins. And this was an extraordinary tour de force. I think there's a very strong argument to saying that had Avery died in an operation, a very crucial operation he had 5 years previously, in which he might have died, that the double helix would have been delayed by 10 or 15 or 20 years. I mean, because without knowing it was DNA, we wouldn't have known, people wouldn't have gone after it or even understood the importance of it. So this was really the most important experiment. Once this was known, then, the structure of the double helix was going to come out pretty quickly. And we all know that, if it hadn't been Watson or Crick, it had been somebody else within 6 months. And certainly it would have been Franklin if they hadn't stolen her data from her. One needs to say this now. In this day and age, it was quite unconscionable what they did. So I think this is, and that's 75 years also, and this is taking place exactly at the same time as Schrödinger's writing his book. Okay, so here was this wonderful structure, and sadly, the truth was rather prosaic. It was a molecule that seems to obey the laws of chemistry and physics, and there didn't seem to be any magic, except for the sort of the beauty of the chemistry. And Peter has described that wonderfully before me. And we've also discussed why did Schrödinger not reply to Crick's letter, and I think we've also heard the answer. I think he was basically disappointed. I think that probably was the answer. The key problem then is for life, for this process of reproduction, is the DNA which contains all the information to make all the enzymes that will create the cell. And really, the DNA is the only essential component of the cell. You can take away all the other components of the cell. You could take them out one by one, and the cell will still survive, because as long as the chromosomes are there, they can remake this stuff. It's the only essential component of a cell. And what you've got to do, obviously, is to replicate it, but you mustn't just replicate it. You've also got to segregate it. So propagation of DNA requires A, replication, And the mechanisms of replication, broadly speaking, emerge from the structure of the double helix. It was immediately, it has not escaped our notice. Bloody obvious, wasn't it? So now, the actual enzymology of that process has proven to be very complicated, and we're only starting to really, but the basic mechanism emerged very simply from that structure. But the segregation process, this business of taking the sister DNAs and moving them to opposite sides of the cell before it divides, has proven to be a much more complicated problem. And I hope I'll try and explain why in the rest of this talk. So my homage to Schrödinger, and I guess my argument a little bit with Peter, is life is not just chemistry. Because it turns out that the process of chromosegregation concerns physical principles. There are chemical ones that are involved, but there are physical ones that actually don't involve much chemistry, and that's why it has been such a difficult problem. If it's a chemical problem, you can measure the damn thing, because there's a difference between the two molecules, you can measure them. But if there's only a physical change, namely topological, it's very hard to measure, and that's why it has been proven so difficult to solve this problem. Anyway, this is therefore my homage to a physicist, that actually the segregation problem is largely about physics. Now we've got to go back to the double helix, and one of the first things that Delbruck said when Watson told him about this structure, he said, well, we have a problem. How do you replicate it? Because you've got to unwind this stuff in order to make according to the double helix, you have to unwind the two strands in order to make the two copies. How do you unwind it? You then are going to have this enormous topological problem. And so at the time, largely people kind of felt that they thought that the DNAs were actually chromosomes were actually made up of lots of little segments of DNA that were joined up by little linkers. They'd had no conception that a chromosome was one molecule of DNA from one end to the other. And if they were smaller, then you largely minimize the unwinding problem. It's much easier to deal with. And that was, Delbruck wrote a little paper on the basis of that. And here's an example of what people were thinking in 1958 of the structure of chromosomal DNA. I've got to take issue with one thing Dieter said. Herbert Taylor did these wonderful experiments that proved that replication was semi-conservative, as predicted by the Watson-Crick model. But the one thing he did not conclude from his experiment, which he should have, which is what Dieter concluded quite rightly, is the only other way of explaining their data was that it was also a single molecule. And it is absolutely wonderful. Every student should be made to read this paper. It is the most underinterpreted paper ever written in biology. Because they were right, it was semi-conservative, but they should also have concluded that each chromosome is a single molecule, and they did not. And they came up with a fantastical, complicated model of how you could have, where it was made up of lots of different molecules, but how did they all end up in one chromatid? It's wonderful, the mess they got themselves into. So anyway, that's how people thought about it in 1958. Really, the penny only really started to drop when people started looking at, basically with electron microscopes, this absolutely dramatic picture of DNA spilling out of a bacterial cell from Kleinschmidt in 1961, and they looked at this and say, wow, there are no ends, and I can see basically, they couldn't see any ends and concluded that probably it was a single circular molecule in bacteria. And then there's this wonderful autoradiograph of Cairns in 1963 looking at the replication of the E. coli chromosome. So around the early 60s, the penny dropped that actually each chromosome was a single DNA molecule. It took a while to work out that it was true for you and me as well, but that was in bacteria. And that is what happens when you do the equivalent experiment with say, a human chromosome. I've blown this up. There's the double helix, here's the DNA, and there's all this DNA here. It's a fantastic amount of DNA. And really the topic of the last part of my talk is how on earth do you get from this to a chromosome, given that you've got to organize all this stuff. Now, to give you to give you a metaphor to really understand the nature of the problem, I actually have a second job, which fortunately Dieter didn't mention in his introduction. I spend quite a lot of time in the south of France making wine, and you buy a vineyard and it's in terrible shape, and you've got to rip out all the trellising system because it's all tangled up and broken, and you replant vines and everything it is. Well, you don't take out much wire, and it ends up with a tremendous tangle. Now, just imagine if DNA was not DNA, but it was one of those wires, okay? Now, a wire like that is, say, 2 millimeters thick. Now, DNA is 2 nanometers thick, so that's a millionfold difference. Now, in one of our chromosomes, in a human being, just one of our chromosomes is roughly, is about 5 centimeters long of DNA. So if it was such a wire, it would be a wire like this. This is maybe a few hundred meters. It would be 50 kilometers of that wire just in one of our chromosomes. And that's you and me. The poor old Mexican axolotl, which has got no more genes than you or I, but somehow it's accumulated more DNA. Each one of those chromosomes would be 1,000 kilometers long of wire. And yet the mechanism by which this is organized into chromosomes is completely scalable. It doesn't mind how much DNA. So whatever mechanism is being used, it doesn't matter on what scale. It'll work on a tiny chromosome or an absolutely enormous one, and the thing still works. And this is really mind-boggling. How do you organize that into a chromosome? Dieter pointed out, the fundamental problem, but that really gets you thinking. Oh dear. Apologize. You mustn't press them both. Okay, so the first point that I just want to point out about this replication business, not only have you got to sort of organize that wire into something that won't get all tangled up inside the cell, but in addition, when you replicate it, you've got to separate, at some point, you've got to separate all the tangled wire. You've got to separate one 50-kilometer long tangled wire from another one, namely the 2 sister wires, okay, or the 2 sister DNAs. And this actually gets more complicated still, because when you replicate, you have to open up the DNA in order for DNA polymerases to replicate the DNA. And to do that, you have a helicase that, as it were, is forcing the DNAs open. And now, as it does that, is it's charging down the chromosome and it's unwinding the DNA. Well, what happens? If you were to what happens? Because it's each 10 base pairs will be a whole supercoil. It's generating a wave of positive supercoiling ahead of the fork. Okay, as you're unwinding the wire. And so therefore, one of the things it does is the replication fork recruits topoisomerases, particular the topoisomerase 1, whose purpose is to remove these positive supercoils, so making sure the wire doesn't get tremendously tangled up in front of the replication fork. But that's one way of dealing with the positive wave of positive supercoiling. But another way is that simply what would happen is the fork just will twist. Okay, and if that happens, of course, then the sister DNAs after replication end up being wound around each other. And it turns out that when our chromosomes get replicated, most of this positive supercoiling that's generated is indeed resolved by topoisomerases. They just make essentially a nick in the DNA, and so it creates a swivel. But some of it gets turned into this intertwining. So those sister DNAs after replication are also intercatenated or intertwined. And so you've got this problem. How on earth do you separate these two things before a cell divides? Is that clear? Yeah. I mean, it's physics, isn't it? It's not chemistry, right? Is that chemistry? Okay. So here we go back. The interphase. It turns out the chromatin is not just all over the place. It turns out that in interphase there are chromosomes, as Boveri first predicted. They occupy a larger space, but they're still highly organized. But then when the cell goes into mitosis, they assemble into Fleming's Faden, which we now call chromosomes, and we now know that these two, they're called chromatids, and it is one sister DNA from one telomere to the other, and that's the sister DNA from one next telomere to the other. Okay, so somehow this gets reorganized into this, ready to be segregated. But actually what you can actually see is the heavy lifting of segregation is done by the chromosome itself. We'll come to in a moment. Later on, the cell has to pull on the sisters and pull them in opposite directions. But basically, the sister DNAs are pre-segregated by enzymes that are working on the DNA itself, and that's what we're going to be talking about. So here are the problems. When you transform this mass of chromatin and build a chromosome, and people refer to this process as chromosome condensation, which is a very unfortunate word, because as physicists, you all know what condensate means. A condensate is an equilibrium state that will depend on temperature and pressure and so forth, and it literally will be condensation. And chromosome condensation is not like that at all. In fact, it turns out that the enzymes that so-called condense chromosomes create these structures are actually working against the tendency of chromatin to form condensates. So it's a total misnomer. What it does, though, is it takes that tangled mass of DNA and a tangled mass of sister DNAs all tangled up together and somehow assembles one sister DNA into this chromatid and the other sister DNA into this chromatid, and you end up with this sausage shape as opposed to an agglomeration. And that's the first mystery. How do you explain the cylindrical structure of chromosomes? The second question is when later on, when microtubules will then pull on the centromeres and actually move the sister DNAs to opposite sides of the cell, you don't want the whole chromosome to, rather like a ball of wool, to unravel when you pull on it. It must have a certain cohesion along the axis of the chromosomes. You start pulling on it, it mustn't just fall apart. It must have a cohesion or a stiffness or elasticity. And these things can be measured by physical means. And they don't just unravel. So what confers this longitudinal stiffness or elasticity, which is fundamental? Lastly, and this is really the most mysterious thing, is how is it that the sister DNAs segregate out, individualize into separate chromatids? How does this end of that very long DNA molecule know that it's to be assembled into the same chromatid as this one? And remember the wire, it's 50 kilometers. In the case of the axolotl, it's 1,000 kilometers away. How do they know they're on the same molecule? Okay? It's remarkable how few textbooks, if you go back 10 years, almost none of them sort of mention these problems. But anyone who's a climber, it's obvious. Anyway, and the last problem is that, yes, you've got to segregate the sister DNAs into separate chromatids, but they must, rather like dancers of the Wiener Waltz, they must stay, in order to dance, you've got to be connected. I mean, real dancing, you've got to be reconnected, okay? The reason why you've got to be connected is if after the segregation they were not connected, how would the cell know how to pull one in one direction and the other in the opposite direction? But only if you're connected and you pull in the right directions and they remain connected, you feel the tension, then you know you're pulling in the right directions, okay? So therefore you have to have sister chromatid, a little bit of something holds these chromatids together to ensure that you can then pull them in opposite direction. Again, it's physics. Okay, and this is the process. These microtubules will attach to proteins that are associated with particular parts of the chromosome. And if you pull in the sister DNAs in opposite directions, and only if they're still held together will you generate tension, and that tension stabilizes these connections. So that's how you get getting them pulling in the opposite directions. So here is the fundamental problem. You start off with this. How on earth do you build that? How are DNAs woven into chromosomes? That is the fundamental problem. This is a lovely picture from an ex-student, ex-postdoc of mine, Raquel de Oliveira. Okay, so from chromosomes to DNA, only half the story. Now we've known from work that started off, well, we've known for a long time that most of the DNA in our chromosomes, albeit not in bacteria, is folded up it's associated with histones, the so-called nucleosome. There's two wraps of DNA around each histone octamer. And this plays a role in organizing the DNA to some extent. It plays a very important role in neutralizing the DNA. And because these histones also have unstructured tails, those tails are subject for enzymes that can modify them and therefore recruit other proteins and so forth. But actually, the nucleosome, it turns out, doesn't have an enormously important role in the structure of chromosomes. And one of the reasons, but nevertheless, for about 35 years in the textbooks, this was the model for how chromosomes were built out of DNA. This was in the textbooks for about 35 years. Right, Dieter? Is that correct? Yes, this part of it is correct, but this part was fantasy, okay? And it's probably completely wrong, but the key point was that was in the textbooks. People were telling generations of students, this is how you built chromosomes. And I always say to my students, it's a totally blindingly simple thing, is any theory or model, I don't like the word model, the only word I like is explanation. Can you explain something? And to me, this people called a model, okay, but it didn't explain anything. The properties of the nucleosome does not explain how you get from here to here. End of story. No explanation. No model. But you don't need the word model. I suspect physics has the same problem. There are a lot of models out there that aren't explanations. Okay. Well, here's a beautiful experiment that was only done a couple of years ago by Tatsuya Hirano. He actually managed to assemble sperm. You can get DNA out of a sperm and put it in an extract from a frog egg, and the frog egg will assemble that DNA into chromosomes, and it does a very nice job. But actually, you can deplete the nucleosomes from those, or you can prevent the cell from incorporating nucleosomes onto that chromatin, and still that extract will assemble the DNA into chromatid-like structures, okay? And this was a beautiful experiment. We kind of knew this would be the case because we know bacteria have the same problem. They've got exactly the same problem as we do, and they don't have nucleosomes. But this was a wonderful demonstration that you can build chromatid-like structures without nucleosomes, okay? It was a lovely experiment. Now the next, so then there was the next thing was, well, if it's not nucleosomes doing something magical, what is it that builds these cylindrical structures? And here we go back again to Walter Fleming. Within a couple of years of him describing mitosis, he described these wonderful chromosomes from meiotic cells where the egg is growing, and so these cells they're pumping out stuff. They're being transcribed. They're making all the stuff that's going to be necessary for development, for the embryo to get to a point where the fish has got a mouth that can feed itself. So it has to have everything there, and so there's an enormous amount of synthesis. And because of that, you can then see under the light microscope that the DNA of the chromosomes has a loop structure. You're not actually looking at the DNA, you're looking at DNA which is covered by RNA polymerase transcribing it, making the RNAs will then be turned into proteins and so forth. But basically, by looking at these kind of chromosomes, you could see this loop structure. They were called lamp brushes by their similarity to the lamp brush. And subsequently, it was shown that these were indeed single DNA. There was a single DNA molecule going around from here to here. So that then led to so in meiosis, it was clear that chromosomes had this loop structure. There was some axis around loops were attached. And then this model was generalized by Uli Lemley and his colleagues, who then suggested this was also true in mitotic cells. Namely, there was a sort of proteinaceous scaffold around which loops were emanating. Okay? Now, you may say this is a nice model, but the problem is it may be a model, but it ain't an explanation. Because the problem is the following, and it is the case that if you isolate mitotic chromosomes and you extract the nucleosomes, you can see there is a kind of proteinaceous axis. Is that the scaffold? But the problem with the chromosome scaffold model is that it didn't really satisfy, for me, it didn't provide an explanation for how you build chromosomes. Because the fundamental problem is how would all the DNAs from a single molecule be attached to the same scaffold? It was the problem of DNA identity. If you have this protein scaffold and you're attaching DNA in loops to the scaffold, how did DNA from one molecule attach to one scaffold and the DNA from its sister to its sister scaffold? And the scaffold hypothesis could never explain that. Okay, so it was fundamentally flawed from the beginning. But more seriously, John Marco showed, he did rather nice experiments to say, if there's a proteinaceous scaffold, he was able to he was a physicist who got new chromosomes out of cells and pulled on them, measured their elasticity. He said, if the longitudinal axis is created by a proteinaceous scaffold, and I go in with a nuclease that cuts every 20 kilobases, nothing should happen. The elasticity should be unaltered. And he found the whole thing unraveled. Okay, so there isn't an autonomous proteinaceous scaffold. It was a very nice, very simple experiment done by a physicist. Okay, so we have a problem. We do not have an explanation how you get from DNA to chromosomes. And here we've already mentioned François Jacob, and somebody else has essentially already mentioned what essentially Jacob said, that the process of experimental science does not consist in explaining the unknown by the known. It aims to give an account of what is observed by the properties of what is imagined. And to me, what this is saying is that the driving force of science is realizing that current explanations do not suffice and that you have to come up with something new. And that was, in fact, what Newton did. That's the birth of modern science. Magic is at the heart of all science. The birth of physics, the birth of all modern science was the law of gravitation. Newton invented a completely imaginary force with imaginary properties, but he said, if it had these properties, I can explain the movement of the planets. And his contemporaries hated it. They say, well, that's cheating. They were all Cartesian. They wanted an explanation in terms of what they already knew, which they'd had for the last 300 years, all these bloody circles. And we still don't really understand Newton's force, if I'm correct. I know some of you in here are probably working on it, but we still don't understand it. But nevertheless, it's got us a long way, and this is what Jacob is saying. And obviously, this is and this is what Schrödinger was. This is why his book was so important. He's saying we don't understand that, and that's why it caught people's imagination. So clearly, we need new enzymes that have new properties to explain all of this. And the enzymes I'm going to talk to you about now are these absolutely remarkable proteins. They're very difficult to work with because they don't have the same kind of structure that Peter described, all these compact alpha helices. They're not actually sort of enzymes in the same sense of the word. Essentially, they are two rod-shaped proteins, about 50 nanometers long, with an ATPase domain at one end and a dimerization domain at the other. They dimerize to form V-shaped so-called SMC proteins. In eukaryotes, they are heterodimers. And then these ATPase domains are interconnected by a so-called Cohesin subunit, one end of which binds to one ATPase, the other to the other. So they create a sort of gigantic ring structure. And then there are proteins that are then recruited to the complex by binding to the Cohesin, and then those proteins are necessary to activate the ATPase, to get it to do work on DNA and so forth. I don't want to go into too much details because we actually really have almost no idea how these enzymes function, but I will tell you as much as we know. And the key point is the enzyme that's involved in packaging DNA into chromosome-like structures in mitosis is Condensation. The one that's doing it in interphase is Cohesin. And here's just a little bit more detail. These are the ATPases, these are the connections between the Cohesin. This is the dimerization domain up here. There are a number of features here which we don't need to go into the details. And the structural biology, as you imagine, is difficult because, but it is coming along, and the field is really, it's a very exciting phase for the field. The key point is that those two ATPase heads must come together, and when they do so, they sandwich a pair of ATP molecules between the heads, and then when they're hydrolyzed, the heads will then come about. So that's the sort of basic enzyme. And somehow this ATP hydrolysis, due to the engagement in the presence of ATP and subsequent hydrolysis, is fundamental to everything these proteins do. And one of the things that we do know is that sort of DNA gets sort of trapped inside, seems to get trapped inside this ring. Now, the other thing is these regulatory subunits also bind DNA, and it'll become clear why one may need multiple DNA binding sites within these proteins, some of which may be ATP dependent, some of which may be ATP independent. But these regulatory proteins, which bind to the Cohesin subunit, do also bind DNA, and these are actual real structures. And so we end up essentially with, this is the nucleosome, this is what the condensin must do, is turn this into this, and you can see that the condensin is a very different beast. Okay? Now how it does this, we don't understand, but it is taking nucleosomal DNA, and this enzyme that has these components, this is just naked DNA, that's nucleosomal DNA, this enzyme is responsible for this transition. How does it do it? Sorry. Well, I got back, Gustav is there. Is Gustav there somewhere? Yeah, Gustav is there. He was my climbing partner. I got back from a long climbing trip in the Dolomites with him. And we normally climb with single ropes, but in the Dolomites you need to retreat sometimes. And so you climb with 2 ropes, but if you normally climb with 2 ropes, you're not used to climbing much with. If you're used to climbing with 1 rope, you're not used to climbing with 2 ropes, and they generally get tangled up. And of course, one of the things that you use when you're climbing also is carabiners. So having got back from this climbing trip, I had my ropes, and we had untangled them, but I was just fiddling around with them and with the carabiners, and realized that actually one way that you could convert that immensely long DNA molecule into a chromatid was just taking a bite in the rope, sticking it through the carabiner, and extruding loop out of this through this ring. And so the idea was that condensin might be a loop extruder, and so it would just start. It would take a bite in the rope, extrude the loops, and they'd just keep going. They're just dumb enzymes. Nobody's telling who to coordinate with what. They just keep doing it until they run into each other. And this is happening in 3 dimensions, and you can basically take the DNA and assemble it into a chromatid-like structure. This was really just a sort of fantasy idea, but it was a genuine attempt to explain how it is that you could create chromosomes. And you can see that the condensin does indeed end up along the longitudinal axis, which was in the scaffold, perhaps, and indeed condensin is the main constituent of the so-called scaffold, but it's not a scaffold in any sense of the same. The longitudinal axis is not something that was built, it's something that simply emerged from the activity of individual proteins. And it explained, how you created a longitudinal axis that had a certain elasticity, it explained why it was cylindrical. It also helped to explain how it is that when you do this process, you separate, you cleanse on the right, here's a simulation of this. This is done by 2 other groups. I'm not a physicist or a molecular modeler, but basically this basic idea, they've shown that in principle it works. But I have a healthy skepticism about modeling. I think if you put in the right parameters, you can generally model anything. You need more, but anyway, the modeling shows the basic idea is fundamentally sound. But what you can see is that obviously most of the DNA, if it's going through this extrusion, will be cleansed topologically, and all of the intertwinings will get focused in this region here where topoisomerases will be focused, and that's indeed where topoisomerase goes and will get involved in decatenating these DNAs. So that's the model from the Miani and Marco labs who've developed this idea way beyond what I did. So basically, loop extrusion solves the untangling problem. It explains how they end up with circular DNAs. It explains how this end of the DNA molecule knows it's on the same as this one. It explained everything. It really did explain it. There was only one problem, and that was, did condensin or cohesin have this property? And last year, there was a beautiful paper in which, well, this will run again, Decker's lab, together with an ex-student of mine, Christian Herring, they took lambda DNA and put it on a glass slide, relaxed, and then added condensin to it. And the condensin was capable in an ATP hydrolysis-dependent fashion in creating loops and extruding them, but because it was extruding them, it ended up as a ball, because that's what the shape that DNA will normally. But if then you flow in buffer, you can actually show that that is a loop. So if we run that again, you'll see this process of, oops. So here's under flow, and here you can see the loops being extruded by the condensin complex. Can you see that? Yep. So what's remarkable is this can extrude DNA at somewhere between 600 and 1500 base pairs per second. This is incredibly fast enzyme. But the remarkable thing is in bulk, or the ATPase is a very weak ATPase. It's only about 2 per second. So either these numbers are wrong, namely it is much more active as an ATPase when it's doing this, or there's something really magical about how it's doing it. But this is a totally unique DNA helicase. Basically, it must be it's a DNA translocase, but how it's working and the step size must be absolutely enormous. And how it does so, we absolutely don't know. So that's what condensin has done. It's creating these structures in mitosis, but in interphase, the chromosomes are actually also organized, and they're also organized into loops by cohesin. Okay, and we now know that both cohesin, and it turns out that Jan Michael Peter's lab here in Vienna showed that if you just prevent cohesin from turning over on chromosomes by knocking out one of its regulatory proteins, that cohesin will assemble interphase chromatin into chromatids in interphase. They're not in mitosis, but they're building chromatids. So cohesin is a loop extruder just like condensin. And just recently, Jan Michael's lab and also a lab in Dresden has shown that if you take frog extracts, it is condensin that will extrude loops, and in interphase, it is cohesin. So they're both very similar, they're related, they're first cousins of this class of enzymes. They're both doing fundamentally the same thing. One of them's doing it in interphase, and the other's doing it in mitosis. Now, so that's where we are for how you build chromosomes. You get from DNA to a chromosome. But there's one last property of the chromosome that you've also got to explain, is that you've got to hold these sister chromatids together. And it turns out that cohesin is not just a loop extruder, but it is also fundamental in holding sister chromatids together. In fact, that's how we discovered it when I was here in Vienna. And so I'm just going to talk a little bit about how it does that. This is what happens in a normal cell in mitosis. The sister chromatids are held. You build chromatids, but they remain cohesed, and that allows you to align them on the spindle. And then at anaphase, you break the connection, and then they go off to opposite poles. If you don't have cohesin, they're not held together, and all hell lets loose in mitosis. You cannot segregate DNAs to opposite sides of the cell without cohesin. And here's this, and what happens is that the cohesion remains there until suddenly, when all of them come under tension, then and only then is an enzyme activated, this separase enzyme, and that's what, it's the activity of that enzyme breaks the bond, and the sisters can then go to opposite poles. And what we discovered when I was here in Vienna, there was a very talented postdoc called Frank Ullmann. What he discovered was that what separase does is it just cleaves the Claisen subunit of the cohesin ring. And in a moment you'll become clear why. And that cleavage is sufficient to break the connection. So cohesin is holding the sister DNAs together, and that cleavage is what triggers their disjunction. So this sister chromatin cohesion, Dieter's mentioned G1S, G2. What he didn't mention is that this is this division of the cell cycle into these phases is unique to you and me and oak trees and yeast cells, but it's not true of bacteria. In bacteria, there's no such thing as G2, because basically the segregation process takes place as soon as you start replicating it. And that's because bacteria never develop sister chromatin cohesion, because it's cohesion that allows you to replicate the DNAs, but because cohesin during replication holds them together, then it retains, then they stay together, and they can stay together for half an hour or several hours or a few days. In the case of a human oocyte, the cohesion that holds the bivalent chromosomes together will be generated at birth and will not be used until fertilization. And so it's several decades. So essentially, it's the sister chromatin cohesion that allows you to separate S phase from M phase. And without that separation, we would still be bacteria. I think it would be safe to say. Yeah. And it turns out that obviously things go badly wrong in oocytes as they get older. And one of the things that possibly goes wrong is that cohesion is breaking down. And once you've lost that connection, then obviously it doesn't work any longer. Okay, so in the last little bit, I just want to tell you our work in yeast about how you build cohesion. The discovery that cohesin formed this ring and that separase opened it up led to this model here of how you hold DNAs together. Namely, sister DNAs get trapped inside this ring somehow, and then separase releases them by just cleaving the ring apart. And to test that, we turned to these tiny little chromosomes in yeast. These were just small molecules, which they're still segregated normally like a chromosome. And the cohesin holds them together. And so according to the ring model, or the ring explanation, the DNAs would be held together by a cohesin ring. They're catenated, as it were, by the ring. And so what we said is if we could surgically cross-link this interface, and this interface, that is, take something that's a physical thing and turn it into a chemical thing, then we could then, if we were to sort of purify these and then boil essentially denature all the proteins, if we had turn that ring, which is otherwise just 3 different proteins bound to each other, but if we chemically circularized it in this way, then you could denature all the proteins, but the DNAs would still be trapped inside that denatured protein. And so then they should run as a dimer by gel electrophoresis. So essentially, the aim of this was to do a little bit of chemistry to turn something that's physical into something that's chemical. And because it's chemical, then we can measure it. So that's what we did. We used this chemistry. We had to get structures. This was with Jan Lerver. We had to actually get crystal structures to know where to put cysteines, and then we could use this chemistry using a thiol-specific bifunctional crosslinker to actually surgically put very specific chemical crosslinks to crosslink these 3 interfaces. And if you had cysteines here, here, and here, and you had this crosslinker, then you could take a yeast cell and get about 50–60% efficiency here, here, and here. And so you ended up circularizing about 20% of the molecules. Okay, there's 3 interfaces. And when you do that, and only when you do that, then what you find is there's 2 new forms. So here's all this DNA is associated with cohesin, but this is a form where you've, as it were, got a single chemically circularized cohesin ring that's catenated the DNA, and is therefore, it's slowing down the DNA in the gel by electrophoresis. And this particular band up here represents 2 DNAs that are catenated inside a single cohesin ring that have been chemically circularized. So we called these catenated monomers and catenated dimers, and this is what we believe is cohesion, and we believe this is formed when cohesin just goes onto chromosomes without replication. So this was the basic idea, is that when you build cohesion, you're building these structures, and we can measure these by measuring these catenated dimers using this cross-linking assay. And so the real question is, where do these structures come from? Do they come from cohesin rings that were on the chromosome beforehand, or are they brought in during replication itself? It turned out so here's the fundamental problem. Here's the unreplicated DNA. Here's a cohesin associated with the unreplicated DNA. Here's the cohesin associated with the nascent DNAs that has trapped those two DNAs inside the ring and has formed cystochromatic cohesion. Well, how was this structure built? Was it built from cohesin that was on here in the first place? In which case, how do you convert this to this, and when this has got to pass through here? And this is all to scale, okay? Or is the cohesin loaded de novo? There's obviously some special chemistry going on here, and Peter mentioned the 5', 3' and all of that. So you can recognize these as sister DNAs. Maybe you load cohesin on here, and because you've got the single-stranded DNA, then you can recognize this is the sister, and thereby loaded on de novo. Well, the bottom line, what I'm going to tell you, is that both mechanisms exist. We've been able to measure the transfer of cohesin that was on there before to these structures after replication, and we've been able to find proteins associated with the replosome that are involved in that transition. We call that the recycling pathway. But in addition, however, you can get rid of that pathway, and there's another pathway that involves other proteins associated with the replosome, which are involving loading cohesin on de novo after the fork's just gone past. And both of those systems can build cohesion. And here's very quickly there's a very important regulatory of cohesin called SSE2. It's essential for driving the ATPase, and it's fundamental to get cohesin onto chromosomes. This is measuring cohesin on a yeast chromosome, and if you don't have SSE2, very little on it. But remarkably, this subunit is, although it's required to get cohesin onto chromosomes, and it's required to build these catenated dimers, but once you've built them, and you've got a G2 cell that's holding the sisters together, you don't need it to maintain them, to hold them together. So you need to get cohesin onto chromosomes, but you don't need it to hold the sisters together once you build these structures. So here basically is our question, is there this, can we measure this pathway, can we measure this pathway? And fundamentally, I don't have time to explain how we did this, but we've been able to develop an assay to see these guys turn into these guys, and then we've actually found out that that process does not require any de novo loading. We can get rid of SSE2 and it still happens. And then we can, okay, with that assay, we can then say which of these proteins that are associated with the replosome that are required for this transition. And here then, here's the question. Now we can measure this process. Are any of these proteins here necessary for this transition? And here it turned out that some of those replosome proteins are actually not essential. You'd think if they're required for replication, they should be essential, but they're not. They're not essential for replication, but they do also have modest cohesion defects. And the yeast geneticists have put them into two different groups. And if you took a gene out from this group and knocked it out and combined it with another gene from that group, nothing much happened. They were still had modest cohesion defects. Likewise, this group. But if you combine them, if you, as it were, knocked out a gene in this group or any gene in this group, then the cell was dead. And so the idea was there might be two cohesion pathways, and we said, okay, we think there's two means of building cohesion, either recycling or de novo loading, and now we have an assay for recycling. We can test, is it this group or this group that's involved? And just remember the color coding here, okay? There's the green ones, the green group, and the brown group. And so what we discovered is the green group is involved in the recycling, and the brown group is involved in the de novo loading. And for example, this protein, TOF1, CSM3, CTF4, and CHL, well, those are all involved in this pathway, and this protein here is involved in this pathway. And here's an example. Just this is the sort of experiment. Here's the basically we'd preloaded the cohesin before replication. We'd prevented any more cohesin going on to the chromosome. We'd say, can this be converted into CDs? So this is just DNA associated with cohesin, DNA that's catenated cohesin, no CDs because they haven't yet replicated. And then when you allow them to replicate, can these be converted into these, okay? So these are these ones here, these ones here. Can you produce these? And you can see this happens in the wild type. This particular mutant can do it, this one mutant can do it, but these mutants cannot. You don't need numbers, do you? But you can see these are the green guys. They can't do it, and these are the brown guys that can. And the bottom line is that, okay, the green guys can't recycle cohesin, but if you allow cohesin to be present during the time the replication fork goes through, then in fact, then the green guys can also do it, okay? And so therefore you then asked, well, how are they doing that? If we knock out those who are involved in the de novo loading, that is, if you knock out the green guys and the brown guys, then they can't do it at all. And so the bottom line is that there are these two pathways. And very interestingly, one of the proteins that's involved in converting this to this is this protein TOF1. And TOF1 is the protein that recruits topoisomerase 1 to the fork to remove all that positive supercoiling. Okay, and if you don't have it, you end up with an awful lot of intertwining of the sister chromatids. And whether there's a connection between that and how you get from here to here, I have no idea, but it's all part of the same process. Oops, and I'm finished now. I'm sure I've gone on too long, but I just need to... Yeah. So there we go. The work that I described was done actually by Magoo Srinivas, essentially. The cohesion establishment is what we say in English, a belt and braces job. You all know what a belt is. The Americans call these suspenders. There's a pair there, up there. Okay, it's wonderful that somebody's got some on and they're in full display. Well, in England we call those braces. And when you refer to something being a belt and braces job, it's somebody, you're fixing something up at home, and it doesn't work quite well enough, so you put in another system in, a belt and braces job. It's basically a bodged job. But basically, Cohesin, there's two completely separate pathways to build it, which is why we haven't made any progress understanding this process until we've made this discovery. And the reason why conventional genetics never revealed the genes essential for establishing cohesion, as opposed to cohesin itself that's actually doing it, was because they're not essential genes, because there's these two different pathways. But they are, both of them are essential for meiosis, because all this stuff evolved for meiosis, not for mitosis, in my view. Anyway, here's Magoo here. I don't think he's got either belt or braces on, so life... but life is, as far as I know, is not possible without either one or the other. Once again, homage to Schrödinger. I think he was very influential. I think there are all sorts of things he didn't get right, but I don't think that matters. I mean, most of what we do is done for the wrong reasons. He clearly was influential. Being wrong there's nothing wrong about being wrong in science. In fact, that's part of the whole process of it. And I don't think he made any difference. I don't think Avery was influenced by him at all. And I think Avery was the key person, not Watson and Crick. Okay, it doesn't matter that Crick was influenced, because if it hadn't been Crick, it would have been Pauling. If it hadn't been Pauling, it would have been Franklin. But Avery was crucial, and I don't think he probably read Schrödinger's book. And I guess Avery might have been a chemist. Okay, thank you.
Thank you very much for giving us this picture of the modern developments in molecular biology, which is very, very interesting to see how far we have got. And while we are, of course, rather late, but I think we should allow, nevertheless, one or two questions.
So I have a remark to that, and please stop me if I go too long. There is something, if you have two strands, closed strands, there's something which is called the linking number of the two.
Correct.
And for the two strands of DNA, the linking number must be 0 because you can separate them. But it turns around every 20 steps.
No, it keeps going round, and the linking number is vast.
Yeah, but then the end must rotate when you separate.
Because you have topoisomerases. You see, all you've got to do is nick. For example, the way that type 1 topoisomerase work is they essentially make a nick. And once you create a nick in DNA, it can swivel, and so you can relieve the supercoiling. It is true you don't need topoisomerases if you have very short chromosomes, because the whole chromosome can spin. But for long chromosomes, to all intents and purposes, they're so long, they're infinitely long, and these topoisomerases are absolutely fundamental for this whole process.
Yeah, no, my remark was going, if you then take the next loop, it must go in the other rotation.
No, but it doesn't.
No, it doesn't.
It doesn't.
Okay.
No. And that was Delbruck's problem. He said that when Watson said, here's the model of a double helix, Delbruck said, you've got a problem. And it took another 20 years, at least, before people discovered topoisomerases. But they're one of those things which, when you go sailing or climbing or fishing, whatever it is, you wish you had a topoisomerase. Because the type 1s just create a swivel, and the type 2s allow you to pass one DNA through the other. And the type 2s are essential for to separate the sister DNAs. You need separase to destroy cohesin, and you also need type 2 topoisomerases to allow the catenanes to be resolved. Yeah. There's a good point.
Kim, very, very short remark. First of all, wonderful talk. I enjoyed it very much. The second is, I have to say, I had also physics at the university and I love physics. Yeah. But you gave a wonderful argument for all life is chemistry. In one of your statements, you said, we don't understand, so we have to look for an enzyme.
Yes, yes. No, that enzyme, yes, yes. The enzyme is burning ATP, but it's not modifying chemically the substrate.
Yeah. And
so the substrate is DNA, and it's not, the DNA is chemically unaltered.
Yeah. Wonderful example was also the topoisomerase. I remember weird theories how you get rid of the supercoil, and then it was resolved like the Gordic. You just cut it through, wrap it around.
Yeah, but topoisomerases do chemical work on DNA, whereas these things are just doing, they're physics. They're burning ATP in order to do it. And we really have no idea how. I mean, I've told you basically all we know, but they have these long arms, and that must be, they have these sort of levers. That must be something, must be a crucial part of how they're marching down chromosomes and the way they are. And they're doing so when this is not naked DNA. I mean, it is all nucleosomal. There's RNA, there's all sorts of other proteins there, and they can do it through all of this stuff. It's extraordinary.